Hello students, today we are learn about one of the nanoparticle named as liquid crystalline nanodispersion, and it is also known as cubosomes. The cubosomes having size 100 to 500 nanometer, and these are lipid based nanoparticles. Cubosomes are square and rounded particles with internal cubic lattice visible. Cubosomes are self-assembled nanostructured particles formed by aqueous lipid and surfactant systems. Cubosomes are thermodynamically stable. They are structured like honeycombed. As we see diagram of or the structure of cubosomes, which has hydrophilic drug and hydrophobic drug, uh, which we can incorporate in the cubosomes. Cubosomes are square like or rounded structure which has internal lattices are visible. There are pros and cons. In pros, high drug payloads due to high internal surface area and cubic crystalline structures. The relatively simple method of preparation, biodegradability of lipids, capability of encapsulating hydrophilic, hydrophobic, and amphiphilic substances or drugs also. The targeted drug release and controlled release of bioactive agents. While most of liquid crystalline system transform into micelles at higher level of dilution, cubosomes remain stable almost at any dilution level because of relative insolubility of cubic phase forming lipid in water. Seventh one, the cubic phase of cubosomes can form particulate dispersion that are colloidally and thermodynamically stable for longer time. In cones, challenging large-scale production due to high viscosity of the cubic phase, large-scale production can be difficult. Low entrapment efficiency for water-soluble drugs. Cubosomes are high water content inside their structure, which can lead low entrapment efficiency for water-soluble drug molecules. Limited control over drug delivery. Compared to polymer-based drug delivery systems, cubosomes do not offer controlled drug delivery on their own. Preparation of cubosomes. There are several methods of preparing cubosomes: top-down method, bottom-up method, simple emulsification method. In top-down method, this method involves the dispersion of bulk cubic phase by high energy, which is then colloidally stabilized by using different surfactant polymers. Bottom up method. In this method, cubosomes are formed by self assemble of certain amphiphilic lipids like glyceryl monooleate or monoolene. These are lipids which we can get from esterification of oleic acid. After esterification of oleic acid, we can get the glyceryl monooleate or monoolene. These are lipids in the presence of suitable stabilizers like polyvinyl alcohol, PVA, etc. Simple emulsification method. In this technique involve the simple emulsification of monoglyceride with polymer followed by sonication and homogenization. Application of cubosomes. There are several applications of nanoparticles, which is cubosomes. Drug delivery. Cubosomes can encapsulate and deliver hydrophobic as well as hydrophilic or amphiphilic drugs. Control release. The unique structure of cubosomes offers stability and control release benefit. Targeted drug delivery. Cubosomes can be easily modified with targeting ligands or surface modification to achieve site specific drug delivery, enhance therapeutic selectively, and reducing off target effects. Treatment of skin, hair, and other body tissues. Cubosomes can use to treatment of skin, hair, and other body tissues. Delivery of various therapeutic agents. They are suitable for delivering wide range of therapeutic agents, including small molecules, protein, peptide, and nucleic acids. Sixth one, chemotherapy drug delivery. Cubosomes having employed in chemotherapy drug delivery. As example, we can use as anti-cancer drugs for incorporate in the cubosomes, and we can give to targeting sites of cancer cells. And seventh one, antibiotic delivery. Cubosomes having application in antibiotic delivery. Factors affecting properties of cubosomes. Lipid composition. The types of lipid used to construct cubosomes can significantly affect their properties. Drug chemical properties. The the properties of drug being encapsulated, such as hydrophobicity or water solubility, can also affect the properties of cubosomes. Surface modification. The surface of cubosomes can modify to achieve specific properties such as targeted drug delivery. Environmental conditions. The transformation between the different liquid crystalline organization in cubosomes can be tuned and controlled by changes in their temperature, ionic strength, or pH of the environment. Fourth, manufacturing method. The method, there are several methods as we discussed before, uh, whether it is top down, bottom up approach. We can It can also influence their properties. Evolution of cubosomes. The particle size and zeta potential. The particle size of cubosomes and zeta potential, a measure of stability of cubosomes, are important parameter that often evaluated. Entrapment efficiency, the measure of efficiency of drug being encapsulated within the cubosomes, means how much amount of drug is encapsulated in the cubosomes. We can uh, measure from entrapment efficiency. Third one, in vitro drug release. This test measured the rate at which the drug is released from cubosomes, means how much amount of drug is released from cubosomes. We can measure by in vitro drug release. In vivo evolution. This involves testing of cubosomes or testing of cubosomal formulation or drug formulation in the living organism to evaluate parameter like bioavailability and therapeutic efficacy. Conclusion. Cubosomes are versatile and promising platform for the delivery of therapeutic agents. They offer unique combination of properties, including the ability of encapsulate and deliver both hydrophilic and amphiphilic drugs also. The unique structure of cubosomes provides stability and control release benefits. They can be easily modified with targeting ligands or surface modification to achieve site specific drug delivery, enhance therapeutic, selective, and reducing off target effects. In conclusion, cubosomes represents a significant advancement in the field of drug delivery and nanomedicines. These are the reference for the cubosomes. Thank you.